I have always been fascinated by evolution. How nature creates all those different species among us, driven only by the need to survive in different environments, and even under harsh conditions. For example, deep down in the ocean, the pressure is so high that ordinary fish with its muscles and bones cannot really move. Therefore, the best adapted species is Cycrolutus marcidus, which becomes very unhappy if you lift it to the surface. Also mankind has created some environments which are really hostile for any living being. For example, in a nuclear power plant, in the reaction vessel, the radiation is so strong that only this little fellow, the Deinococcus radiodurans, can survive, which almost seems to enjoy the radiation. And also, at Bessie, there are some places which are really unfriendly for life. They have a pressure less than 10 to the minus 6 millibar, and there is practically no water. And I was wondering, after 24 hours of beam time, is life possible under these conditions? And therefore, we conducted a few secret experiments in a secret laboratory at Bessie, which I will now reveal to you. Okay guys, I'm going to share a really big secret with you. Can you please give me some astonishment? Do some Okay, let's try it again. And next I will reveal to you the secret labs at Bessie too. Thanks guys. So, when you enter Bessie, usually you go through this door into the experimental hall. And then next you open the door, you pass through the door, and then you always turn left to get to your experiment. But you never ever turn right, because it's forbidden. And if you actually do, you will hit this door, and there is a sign on the door, and the sign makes it quite clear that you should not enter this laboratory. But if you happen to have the secret key, and you can open the door, then we will find a laboratory with a few beamlines. It's actually a very cozy lab. And maybe you can see from here that there is a Pilatus X-ray detector installed in this laboratory. So, and this is our experimental setup. We have a light source, we have a diffuser to make the light isotropic in all directions, then we shine it onto a plane and we look at it, we detect the light. This is called a lamp, and you have it in your living room. We do the same, but because we are mad scientists, we use Bessie 2 for the light source, 100 million euro. We have a piece of glassy carbon for the diffuser, around 1,000 euro, and a Pilatus detector which costs 500,000. And if you want to repeat our experiments, approximately 100 million euro you need. Now our experimental results. Basically, when you switch on the lamp and look at the floor, you will see some lit surface, isotropically lit. This is what you also expected, but actually what you find is this. So there is a very strange pattern of lines, some shadows. Ignore the white lines, these are just bad areas in the detector, but the black lines, they are real shadows. And how can this be explained? Well, once you know it, it's very easy. We very rarely dust off our beamline. So the explanation is just this. There is a spider sitting in the beamline, and what you see is the shadow of the spider's web. But this model has still some problems. For example, usual spider silk is transparent for X-rays. How is it possible to see the shadow of the spider? Well, clear. The spider makes a web out of metal wires like lead or copper wires. And this is possible because a regular lab spider got trapped in our beamline, we think, and was mutated. There's a high level of X-rays radiation, and this triggered the mutation of the spider to a lead-breathing spider, which we then called Araneus Pilati Blumbotex. That's the lead-breathing Pilatus spider. We then found out that when we change the photon energy or the wavelength lambda, that also, we increase the wavelength, also the web becomes larger. And for this to explain, we made a model. I have it with me. So this model spider, yeah, it has these bubbles here are sensors for radiation, and by this it detects the wavelengths and moves accordingly. And if we do this, use this model to compute the web, you can see on the right-hand side a simulation for different wavelengths. We never call it wavelengths again. And on the left there is the experiment, and you can see that there are really different shapes of the web that are possible dependent on the wavelength. Also, you might have noticed, that at some point the spider sits at the center of the beam. This is at special values of the wavelength, and using our model we were able to calculate exactly these energies, and by this we can actually calculate the energy of our radiation by using the spider. Now there are still some open questions. For example, shall we keep it? And well, of course, because it's very useful. It measures the wavelength of our beam. It's not yet clear, so what does it eat? So it makes wax from metal wires, so probably it eats the bellows. You should really check your equipment next time for spider bites. And you also don't know how it procreates. Maybe it mates during the shutdown, that it travels to the inner storage ring of Bessie. And everything we know about the spider, we have written up in a paper and published last year in Journal of Applied Crystallography. But the referee said, well, you never actually saw the spider, you only saw the web. So he didn't believe we really found a new species. So we just changed the title and said it's some diffraction effect inside the detector. And then they bought it, and now we have a new paper. And if you are interested in learning everything about it, you can also come to the poster 198 tomorrow in the poster session, where I put up the poster with the spider, and maybe you can also get some pets made out of the balloon.